Good afternoon. I'm excited to introduce our next guests, Alex Fotopoulos, Chief Technology Officer, and Dr. William Cho, Chief Executive Officer from Passage Bio. The format for this session is a roughly 25-minute fireside chat, and if you have any questions during our conversation, feel free to type them into the question box underneath the video player, and we will try to incorporate them on your behalf. And without further ado, Will, Alex, maybe you can spend a few minutes and provide us a brief overview of Passage Bio.
Sure, Matt. I can handle that. So for those of you who don't know, Passage Bio is a clinical stage genetic medicines company. We are focused on developing therapies for patients with life-threatening CNS disorders. We have a pipeline of gene therapies that includes 2 lead clinical stage product candidates in disease areas for which there are really no approved treatment options. So we have PBGM01 for the treatment of GM1 gangliosidosis, and we have PBFT02 for the treatment of frontotemporal dementia with the GRN mutation. For us, 2023 is a really exciting year. We have ongoing phase 1/2 trials for both of these programs, and in 2023, we will be sharing new clinical data from both programs. So for GM1, mid-year, we'll be sharing data from the 7th and 8th patients treated in our phase 1/2 trial. And for FTD, in the second half of this year, we will be, for the first time, sharing clinical data from our ongoing FTD trial.
Great, yeah, lots to look forward to.
Definitely.
How would you define your manufacturing and CMC capabilities and strategy at Passage?
Thank you, Matt, first for the opportunity to be here and discuss some of the CMC and manufacturing aspects at Passage Bio. So in order to kick off sort of the discussion and actually your question, starting from your question, it's important for the audience to know that manufacturing strategy is not something that stands on its own, but rather it's integrated with both the clinical development strategy and the company strategy. The manufacturing strategy is a sort of an enabler of what, in order to get some of the data that Will was talking about, how to move these programs from the preclinical stage to the clinical stage, and hopefully advance them in order to provide the hope we believe we can provide to the patients that we're trying to serve. In terms of what the manufacturing strategy is for Passage, I would try to describe it as a sort of a, with the pillars that form the strategy, if you want. First of all, the manufacturing technology. We're using a very flexible triple transient transfection platform that allows very quick transition from one program to the other. It's very fast from preclinical to clinical, and also it's been a very well accepted platform from a regulatory perspective, which allows to enable the clinical development of our products. We are using long-standing relationships, external relationships, for not only the manufacturing, but also the supply of our therapies. I want to remind the audience that we are conducting global clinical trials on both programs that we having in the clinical development. It's very important to get the patients to support the programs, and especially in the rare disease space, you have to go where the patients are. So you have to do global clinical development, and we're trying to reach all the patients where they are globally in multiple different geographic locations. The last but not least component, which is the more important one and which is the glue that keeps everything together, it's really the talent and the internal capability, having the scientific and the technical team in order to enable everything I said, not only how to run and develop and manufacture these therapies, but also how to engage with regulatory authorities, how to advance the therapies, and how to advance also the technology as we are trying to advance the assets to commercialization.
Understood. And I guess building off that, in terms of your interaction and relationship with the gene therapy program at Penn, how does that relate with to Passage and your manufacturing?
Yes, first of all, it's very important to highlight that we have a very strong relationship with the GTP and Jim Wilson's team and Jim Wilson himself. We are working very closely with GTP in terms of the various clinical assets we have, or at least the two advanced assets we have, plus also some work we are doing in the research and discovery. And that relationship provides, if you want, us with the foundation, the technical and scientific foundation. We have access to a lot of serotypes, and as a matter of fact, to novel serotypes. We have access to vector engineering technology. In a more simple way, the design of the nucleic acid payloads, and also the associated regulatory components that come with them. We have access to the delivery of these vectors. And last but not least, we have also access to many aspects of if you want, the technology itself, not just simply the manufacturing technology, but technology associated with AAV and gene therapy.
Right. And then I guess what other partnerships does Passage have, and how do they relate to your manufacturing, your manufacturing and larger goals as a company?
Sure. Besides UPenn and GTP, we have a long-standing relationship with Catalent that for the manufacturing of our vectors. We have also a number of other relationships as they pertain not only sort of in the raw materials that go into the manufacturing, you ask about manufacturing, such as plasmids, but also how we distribute our materials and clinical supplies by using third-party logistics companies that they have a reach to a lot of countries outside US as well. But I would add also, we have certain relationships with CROs. And the CROs, as much we are talking about manufacturing, there is an integrated component when you come into regulatory interactions and global regulatory interactions, or when you are thinking about testing for some of these clinical supplies. So we are working, we are having a very sophisticated and elaborate network, if you want, of third-party relationships. All of them, we are trying to maintain very strategic relationships, such as the GTP I mentioned. And we are, relying on these relationships on how we work over the years now as we have entered into the clinic. Because not only we know what they do, they know what we want, and that has created the sort of the synergies, if you want, to enable faster delivery of the clinical supplies to the patients, but also more efficient operations for the CMC in general.
Got it. And maybe, do you mind expanding a little bit on Passage's decision tree when deciding which elements to internalize as opposed to which to advance together with partners?
Of course, I would say, the first thing that comes very strong about the choices that you have to make, or at least the drivers on our side about decisions about internalizing versus maintaining external, is first know-how, IP. When there is a critical component of that, we are trying to maintain it internal. I would say also complexity. The manufacture of gene therapies are very complex. Manufacturing is not only just the making, but it has to do with the testing. It has to do with, like I said, getting approved these therapies in US and outside the US. So those components that they are basically more complex, such as potency assays, for example, very complex in the gene therapy space. These are the ones that we are prioritizing to internalize. Also, although we may not prioritize, the opposite of that, when it's a simple assay or a simple operation, we look into what may be a bottleneck as we look into the advancement of the therapies. So when we look holistically, the end-to-end supply chain that I said earlier, key bottlenecks that we feel that we need to overcome in order to enable the delivery of these therapies to the patients, we're trying to either internalize these operations or find ways to mitigate or reduce these bottlenecks by, if you want, not only internalizing, but maybe using a hybrid approach that we can do it better and faster.
Now, from your perspective, how similar is manufacturing of AAV gene therapies to biologics broadly? And, do you have any lessons learned from biologics that can inform AAV manufacturing?
Yes. So, I think you're probably gonna know my answer here. There are similarities and there are also differences. If I can try to keep it in a very, very short and simple way to describe it, when you're thinking about in vivo gene therapy, a lot of the processes and the bioprocesses required to manufacture the vectors are very similar with biologics. And manufacturing of enzyme proteins, or even more, people are more familiar with monoclonal antibodies. There are also big differences, and the difference is really the chemistry, if you want, the biochemistry. In the protein world, you are dealing also only with a protein, an amino acid, if you want, sequence, probably with some post-translational modifications, sugars that you may have. But in the world of gene therapy, it's not only the protein, which is part of the capsid of the vector, but also the nucleic acid payload. So you are dealing both with proteins and DNA. And those things create a lot of interesting challenges in terms of, and differences, if you want, with mAbs, in terms of how they're being manufactured, how they're being produced and purified, and also how they're being tested.
Got it. Now, I understand that Passage uses HEC-293 transient transfection system in your manufacturing. What are the advantages to the system?
Sure. I think I mentioned already a couple of those. First and foremost, it's very flexible, meaning that we are having a number of different products, and clinical candidates. We can switch manufacturing from one to the other by switching sometimes one, maybe two plasmids at a time. And immediately we can manufacture the supplies we need with very minor, very small changes in production records, or even in the testing, because it's all platform testing that we're using for any of our products. It's very fast, meaning that we can transition very quickly from an early discovery candidate to preclinical candidate, and eventually to clinical, and I would add also commercial candidate, because the platform is very flexible in that way and gives us we can advance the platform quickly. Another sort of benefit here, I would argue, is also the fact that has been well accepted from a regulatory perspective. The platform we're using has been approved previously for at least one or two programs, depending on how you define the adherent platform. So that gives us also the confidence to advance the assets and also establish the platform in a way to meet the end goal for any gene therapy company to get into commercialization.
Excellent. And I know that you touched on this a little bit earlier, but where do you see Passage differentiating or applying itself in the analytical methods used to evaluate yield and purity of the vectors that you're making?
We are, for one thing, that what makes, I would say, Passage a little bit different in the analytics is the fact that from the beginning, even when we're going into an IND, we have a potency assay. That's not the case with a lot of programs in the clinic, or even advanced gene therapy assets. And when I say we have a potency assay, I want to clarify we're using an in vitro potency assay, where it's more common to see early on in vivo tests, and eventually companies heading into in vitro. That that analytic, along with a number of other assays we're using, it gives us the strength, first of all, to interrogate the vector from a quality perspective, because quality is very important for the patients here, but also from an understanding and also from sort of the data we are presenting to regulators with our analytics. It gives us the strength and the ability, the confidence when we have interactions that we do know how the vector performs. We do know what is the quality attributes that they are important, and we know what is basically the quality we produce in manufacturing scale and is going to be delivered to the patient.
No, that's excellent. How many people do you have on your manufacturing side right now, manufacturing side of the business, and what have been the challenges in securing the talent there?
It's interesting, let me start from the second one because it's very important. We have been very successful, and we have not only a great talent on board, but also we have been able to retain that talent, and that talent has developed. So we have a scientific talent that now has a lot of experience with our vectors and with also all the programs, and that enables us to do a lot of interesting things, in terms of how we advance the assets. About, I would say, half of the organization in the company is engaging with CMC operations, which include, first of all, the technical team, manufacturing, supply chain, but also development, which is process development and analytical development, and last but not least, also QA operations.
Now, how would you say that the current financing climate has impacted your manufacturing priorities?
I appreciate the question. This is a challenge that we have been going since 2022. Considering the financial headwinds, we have not been, untouched. And CMC, it's a big challenge. You need to appreciate CMC, it's a big spend. It requires infrastructure, requires a lot of spend in advance because of the long lead times required. So in this environment, it has been a significant challenge how to balance, if you want, the needs of the programs, the future needs of the company and the programs relative to how to manage the spend and how to enable the overall company to proceed in obtaining clinical data from our two advanced clinical assets. So that I would call it active management of conflicting priorities. It's been the biggest challenge. And I would say this is also the challenge for a lot of companies in gene therapy and in rare disease space these days.
Mm-hmm. Now, as for scalability of your manufacturing abilities at the moment, is Passage positioned to handle commercialization of your current assets, and should they reach market, and if so, for how many products?
Thank you. I need to start saying that right now and from the beginning, that was sort of one of the strategic directions. We are running at commercial scale our manufacturing platform. We are running in an environment that is also very close to commercialization, meaning the GMPs or all the systems surrounding the manufacturing platform. So even though today we manufacture clinical grade, if you want, supplies, all of that is done at the commercial scale. All is done with procedures that they follow commercial grade manufacturing. And also I may add that since we have been doing that, we have got a lot of lessons learned, and already we have integrated and applied a lot of the lessons learned in the platform. And that's what we continue to do, which gives us sort of the opportunity and, I'm going to use a little bit of a technical term, the CMC readiness to advance these assets very quickly in the different stages of clinical development for all our assets.
Great. And then in terms of your interactions with regulators, how have those conversations been going, and where are you at there?
I would say, from I'll speak first from a CMC perspective because it's a manufacturing. We have a lot of interactions with the regulators for all our programs, and we have been engaging in many communications. In recent times, and this is more important to highlight here for the audience, is that we have very positive interactions with the agency, and not only the agency in the FDA in US, but also with other ex-US agencies. And we have a very, I would say, strong and engaging dialogue. And thus far, we have received very useful and very promising and positive feedback, at least for the two assets that we are advancing in the clinic. So from that perspective, I would call it positive. But of course, when you look into what feedback you receive from agency to agency, there is a variability there for sure. And that's what we are trying to manage when we are looking into where we want to advance the assets in the clinic.
Yeah, I mean, each health authority's got their own expertise, expectations, requirements. The good thing is, as Alex said, we have the experts in-house, so we will work, and we do work with each different regulatory authority wherever they are. One thing that we should bring up is we have heard the FDA talk about hopes for harmonization, and we would certainly welcome any efforts around that.
Absolutely.
Excellent. Yeah. Is there any particular variability between the different regulators across geographies that you can discuss, or?
I mean, there's a lot of things to say here. I would start with the very basic stuff. Sometimes, you have to start from those basic in order to really get an understanding about how some things or many things are different or variable between agencies. Just to remind people, more or less the same number of cell and gene therapy therapies have been approved between the FDA and EMA, but only half of those are approved in both the territories. And we are talking about commercial therapies. In that regard, you can see already there is something that is not, there is some variability and something that it doesn't drive, because everyone wants to get these therapies to the end point. Everybody wants to go into commercialization. So the fact that you have this discrepancy immediately speaks about the variability. And things can start even from simpler ones, like even the nomenclature. Both territories, if you look between EMA and FDA, when they talk about the same topics, they use a different nomenclature. And now when you start diving into the requirements, they are local requirements that basically have different requirements for the sponsor to meet in order to get into the final approval. So and I try to keep very high level the response here, but as you can imagine, the more you go into aspects of not only the clinical development, but also CMC, which is very technical. You might find, for example, specifications, safety, a lot of those things to be different. And as Will said earlier, a big factor here has to do with the experience, the exposure, and the understanding of a lot of the reviewers, but also what are the local requirements, as I said earlier. And those create the variable feedback that you may get from the regulators. Got
it. See that we're close to time, but I do want to ask, what do you see as the biggest CMC challenge in gene therapy right now? And what important innovations in AAV manufacturing do you most look forward to?
Let me handle the first one, and you do the second one, okay, Alex? So I think the biggest challenge to us right now, Alex alluded to before, it's the financial headwinds in the overall space. So when you're in a market that is so capital constrained as we are now, we're just having to make a lot more prioritization decisions. As you know, we downsized our clinical portfolio at the end of last year, and we made the difficult decision to stop 2 IND-ready programs and only continue with 2 clinical programs. Similarly, we have to make difficult decisions in CMC investments. We still need to make investments that are going to enable long-term success, but we really need to pick and choose with a fine-tooth comb to balance short-term financial needs with longer-term de-risking.
Absolutely. I mean, you know, this is the #1 challenge right now for, I would say, myself, my team, the company, and I would say the whole the field right now is struggling with what Will described as a challenge. And those challenges, they create also you have to understand the horizon in terms of the investments becomes very different in terms of where you invest in your capabilities. We are trying right now to obtain the clinical data. This is basically the first step and the most paramount priority for CMC, to obtain clinical data. And then we are looking into how we need to thoughtfully allow investments for the longer term in terms of where we need to make what investments for when the right time and the appropriate data are coming out of the clinic.
Great. Well, thanks so much for your time today, Will and Alex, and we enjoyed having you at our conference.
Thanks very much, Matt. Take care.